All right, let's jump right in. We are living in this pretty incredible moment where deep learning AI is starting to read the secrets of our own DNA. I mean, it promises to completely change how we understand disease, but, and it's a big but, this amazing power comes with a huge challenge, and it all comes down to trust.
And this is really the core question, isn't it? We have these AIs that could potentially predict diseases and literally save lives. But on the other hand, a lot of them work like these black boxes. That makes it incredibly difficult to trust their decisions, especially when something as important as our health is on the line.
So let's really dig into this because this slide just lays out the dilemma perfectly. The promise is massive, right? AI that can comb through our genetic code and find secrets we've never been able to see. But the problem is just as big. If an AI's decisions are totally opaque, how can we possibly check its work? How can doctors, how can patients build the trust you absolutely need for this stuff in healthcare?
So today, that's what we're going to unpack. We'll start with the black box problem itself. Then we'll get into the three main challenges researchers face when they're building these things. We'll look at the different AI architectures they use and the strategies they have to make them more transparent. Then we'll wrap it all up by looking at where the field is right now and what the future holds for trustworthy AI in genomics.
Okay, first up, the genomic black box. Let's really get into why in medicine, just getting the right answer from an AI is only half the battle. We have to know how it got there. You know, the whole reason we even use these super powerful models is because they can see things that we just can't. The links between our genes and diseases aren't simple, they're not linear, they're unbelievably complex. Deep learning can spot these really subtle, hidden patterns in just mountains of genetic data, making connections that would be pretty much impossible for a person to find. Now, it is so important to get this next distinction. Explainability is about understanding why a model made one specific prediction for one specific patient. But interpretability, well, that goes deeper. That's about understanding how the model works at a basic fundamental level. The thing is, all interpretable models are explainable, but it doesn't always work the other way around. Okay, so with that challenge in mind, how do researchers actually go about building an AI that we can trust? Well, it turns out they have this really practical roadmap that zeroes in on three core design choices. And this is the roadmap, right here. It's a three-step process. First, you've got to really deeply understand the data you're working with. Its quirks, its challenges. Second, you have to pick the right model architecture for that specific data. And finally, you choose a strategy to interpret that model's decisions, to basically make that black box see-through. So let's focus in on that second step, the architecture. The actual structure of the AI model is a massive piece of this whole puzzle when you're talking about interpretability. Let's look at a few of the most common types. First, you've got the multi-layer perceptron, or MLP. You can think of this as the original gangster of neural networks. You've got neurons set up in layers, and every single neuron in one layer is connected to every single neuron in the next. It's the original workhorse of deep learning. Then there's the convolutional neural network, or CNN. These were first built for image recognition, and they're fantastic at finding local patterns, like the edge of a face or the shape of a car. Well, in genomics, they're used to scan along a DNA sequence to find important repeating patterns or motifs. Same idea, different data. And this one is really fascinating. It's called a visible neural network, or VNN. These are actually designed from the very beginning to be interpretable. Instead of just connecting everything to everything, they build existing biological knowledge right into the AI's structure. That way, only connections that make biological sense are even created. Each little neuron can represent a real thing, like a gene or a biological pathway. It's pretty cool.
Okay, so you've picked your architecture, you've trained your model. Now what? Well, this brings us to step 3, interpretation. Researchers have a whole toolkit of different strategies they can use to peek inside that black box and figure out its logic. So broadly speaking, these strategies fall into two camps. The majority, about 59% of them, are what you'd call passive or post hoc. That just means the interpretation happens after the model is already built. The other way to go is active, which is where interpretability is built right into the model's design from the get-go, kind of like with those visible neural networks we just talked about. Now, inside those passive methods, the most popular ones are called attribution methods, and they're all working toward the same goal, to give an importance score to each piece of input data. So for example, if a model predicts a high risk for a certain disease,
An attribution method could tell you, hey, gene A contributed 30% to that risk score, but gene B only contributed 2%. It actually shows you the evidence.
And these explanations can have a really different scope. A global interpretation helps us understand the model's general logic across a whole population of people, which is fantastic for general biological discoveries. But a local interpretation, that's all about explaining a prediction for one specific patient. And that is absolutely vital for personalized precision medicine. So now let's look at the data and see where all this research is actually being put to use today. Who's using these techniques and what are they using them for? The first thing you'll notice is that this field is just exploding. I mean, think about this. The analysis this whole explainer is based on found that a staggering 94% of all the research papers on this topic have been published since just 2017. And there's been a huge spike just in the last couple of years. It's moving fast.
So what's the main driver of all this growth? Well, it's probably not a huge surprise. It's cancer research. Oncology applications make up almost half of the entire field. Neurology and immunology are also pretty significant areas of focus.
And what are they actually using it for? Well, 78% of the time, the job is something called supervised learning. That's just a fancy way of saying they're training the AI on data that already has the right answers. You know, like a big data set of genetic profiles that are already labeled high risk or low risk. It's like giving the AI a study guide to teach it how to do a specific job, like classifying a tumor or predicting how a patient will do.
So finally, let's look ahead. Now that we kind of get the tools and we see what the landscape looks like, what does the future hold for trustworthy AI? And what are the big hurdles we still need to get over? One of the biggest takeaways from all the current research is this: don't just rely on one explanation. Different methods have different strengths, different blind spots. The consensus is that using a combination of methods is really the best way to get a solid, clear, and consistent picture of what the AI is actually thinking. But some major challenges are still out there. Researchers need to make sure their interpretations are stable and repeatable every single time. They also need to build AIs that can somehow account for the gaps in our own biological knowledge. And they have to develop totally new ways to interpret the next generation of huge models like transformers. Yep, the same powerful architecture that's behind tools like ChatGPT. And all of this leaves us with a final thought. As these incredibly powerful AI models learn to read the very source code of our biology, the challenge of making sure that we can read their minds becomes more critical than ever. Transparency, it turns out, isn't just a technical nice-to-have. It's a fundamental requirement for the future of a medicine that we can all truly trust.